It's really good to hear about these two diseases. With all due respect to my real life twin boys, hepatitis C and HIV are the real twin loves of my professional career. And so it's a real pleasure to be here and give y'all an update on some new exciting things that are going on in HIV. So Dr. Vierling gave us all the very exciting data that's going on with hepatitis C, and sometimes HIV seems rather boring, that it's not as exciting as hepatitis C. But actually, there's a lot of new stuff going on with hepatitis C, and it actually is fairly interesting, and HIV continues to advance, even though the vast majority of patients are doing well and well controlled. So let me figure out the technology here. It's also good, I wanted to say, to see some old friends I haven't seen in probably 10 or 15 years, and it just reminds me that I really need to get out of clinic more and come and see people. It's really nice to see people. Okay, so my objectives as we finish up tonight are two. One is to just describe the characteristics of new antiretroviral agents that are in development. Some have already hit the market, some are coming to the market soon. And then to sort of put them in perspective and discuss how those new agents may fit into the current treatment guidelines that Dr. Oquizan just went through. Okay, there's 5 trends in HIV development that I want to touch on briefly tonight. One is a combination. So everything's about putting the drugs together. So just like with hepatitis C, when we see these one-pill regimens, and that's the one pill that most people want, they want to take one pill, that's what's going on in HIV too, is that all of the existing medicines are trying to find partners to combine with. And so we'll touch on that just a little bit. Improvements to current medicines to improve on dosing or toxicities. We'll talk about tenofovir alafenamide in quite detail. Development of long-acting agents. So this is depot antiretrovirals. So the concept of giving an injection either quarterly or every 2 months or whatever in order to control HIV instead of having to take daily pills. The development of new mechanism of action. So this is something that is not happening in hepatitis C, at least that I know of, where we're looking for new targets and new ways to kill HIV. Instead of just the old enzymes we've normally targeted. And then finally, no talk on HIV developments would be complete without cure, because Dr. Vierling said we use the word cure in hepatitis C, and we do. We don't use that in HIV, but hopefully before I retire, we will have a cure of HIV. Okay, let's talk about fixed-dose combinations. So Dr. Oquizan mentioned atazanavir cobicistat. So cobicistat, remember, is a new, relatively new, a couple years old now, booster or cytochrome P450 3A4 inhibitor that was developed for an integrase inhibitor, but it's been also used to boost the pharmacokinetics of other protease inhibitors. And so atazanavir, which is a protease inhibitor, been around for 15 years, 10 years maybe, has been combined with cobicistat into a single tablet regimen. And this slide just illustrates the overlapping PK of cobicistat boosted atazanavir with ritonavir boosted atazanavir. If you put this in patients, this is a clinical trial, head-to-head, atazanavir ritonavir versus atazanavir cobicistat. You see comparable response rates on the high 80% rate. This study was reported at 48 weeks, but has been extended to 144 weeks with the same response. So cobicistat atazanavir has been shown to be non-inferior to ritonavir atazanavir. However, one of the general principles and then probably the main principle of still using protease inhibitor in today's HIV patient is the blockage of resistance development. And it's been a sort of a maxim in HIV medicine for the last 10 or 15 years that boosted protease inhibitors, if used against a wild-type virus, at failure do not result in resistance. And would this carry over to cobicistat? Well, this study did indeed show that it does. And so the patients that failed either atazanavir cobi or atazanavir ritonavir, even at failure, had no primary resistance. Whoops. So it's good to know that cobicistat carries that same property as ritonavir when boosting PIs. What about darunavir? It's the other main protease inhibitor that's useful in the clinic. It's also been paired up with cobicistat into a fixed dose combination. It wasn't studied in a head-to-head trial, but rather a single-arm study. And I'll show you the results in a second. I do want to caution you that darunavir cobicistat is only for use in patients that are naive or have a wild-type protease or no protease-specific mutations to darunavir. As you know, in resistant patients, you can use darunavir ritonavir BID, at a different dose, but darunavir-CoB is not studied for that population. So it's only in naive patients or patients with no darunavir-specific mutations. These are the data that show 83% success rate. Again, there's no comparator in this study. The two other bars are just viral load strata. But if you do look at the darunavir study called Artemis, it has a comparable response rate in naive patients. So bottom line, we now have co-formulated protease inhibitors. All three useful protease inhibitors in the clinic now, darunavir, atazanavir, lopinavir, are all co-formulated with a boosting agent. So we've made progress there. There's other combinations that are occurring, 3-TCs being paired with lopinavir. Of course, abacavir, 3-TC, and dolutegravir are paired up. Raltegravir and lopinavir, ritonavir are pairing up. So lots of people looking for dance partners in the HIV world. Okay, what about improving existing medications? The real improvement here is going to be tenofovir alafenamide, or so-called TAF, T-A-F. And tenofovir alafenamide is actually a prodrug of tenofovir. And what it's designed to do is improve upon the renal and bone safety of tenofovir. And also, as a side note, it's also very small milligrams. So it's only going to be 25 or even 10 milligrams, which again allows it for combination, which is the other theme of this talk. So combination with other agents. So this is how tenofovir alafenamide works. So tenofovir, the actual drug that's active, so tenofovir here, active as tenofovir diphosphate, does not get absorbed from the gut to the plasma. So it's, I just got to figure out how to not do that. So tenofovir does not go from the gut to the plasma. So about 12 years ago, tenofovir dipivoxyl fumarate, or tenofovir DF, was the original prodrug. It's the prodrug we have now called tenofovir, or Viriad rather. And so I don't know how to do this. So tenofovir goes into the plasma, but it's very quickly metabolized to tenofovir. Within, it says 0.41 minutes is the half-life. And so what this results is very high levels in the plasma of tenofovir. And then that is delivered to the bones and the kidneys and can produce toxicity. And so tenofovir alafenamide, you can see here, tenofovir alafenamide has a 90-minute half-life in the plasma. And so it doesn't convert to tenofovir very quickly. And so the tenofovir levels in plasma are very low. However, in the lymphoid cells, there's this enzyme called cathepsin A that is responsible for cleaving tenofovir alafenamide to tenofovir into its active agent. And so by doing this, you eliminate the plasma levels of tenofovir and highly concentrate tenofovir in the lymphoid cells. So this has been very useful, theoretically, to lower the toxicity of tenofovir and concentrate the tenofovir in the cells in which HIV lives. And this is a study that shows the antiviral effect of this strategy. In here is the tenofovir 300 milligrams, so that's in dark blue here. And then in this light blue, this is TAF 8 milligrams. So 8 milligrams of TAF had the same antiviral effect as 300 milligrams of tenofovir dipivoxyl fumarate. And so if you go and see the other doses, the doses that was selected for moving forward was TAF 25 milligrams, 10 milligrams when it's boosted by either CoB or ritonavir. This is the PK data that just confirms that tenofovir levels when you're using the tenofovir DF formulation is this high, and then 90% reduction when you use TAF. And so it does indeed deliver on its promise of lowered plasma levels. So what are the regimens that are being developed with TAF? Well, there's going to be elvitegravir, cobicistat, FTC TAF. That is going to be Stribild 2.0. Then you have rilpivirine, FTC and TAF. That's going to be Complera 2.0. And then you have this new combination, darunavir cobicistat, which we said was already approved. And then combine that with FTC TAF, and you have for the first time ever a single tablet regimen complete based on a protease inhibitor. So we've been waiting for this a long time. And so you have a protease inhibitor, boosted protease inhibitor, combined with FTC and TAF in a single tablet regimen. So this is quickly the clinical studies that prove these theoretical benefits. This is a study of ECF TAF. So this is Stribild 2.0 basically against the original Stribild in a head-to-head trial. And what you can see simply is that the virologic outcomes were the same, 90% in both arms. So it worked. But beyond working, did it deliver on its promise to lower bone and renal toxicity? Well, in the spine, you can see here the serial DEXA scans. This is the tenofovir DF arm, okay? So you see this usual decline in the bone mineral density at 48 weeks. But the TAF arm had about half of the decline in bone mineral density. So it appears that tenofovir alafenamide, TAF, has less potential for spine and hip toxicity than tenofovir DF. What about the kidney? This is levels of proteinuria. So in the tenofovir DF arm, you had about some increase in urine protein, and a little bit of increase in urine albumin. But in the tenofovir alafenamide arm, you had less so, even negative. And so what this suggests is that, indeed, tenofovir alafenamide will be safer for the kidneys as well. So it does seem to deliver on its promise of less renal toxicity, less bone toxicity, with the same antiviral effect. Resistance is an issue with the integrase inhibitors. And so what you see here, the ECF-TAF, about 1% of the patients, about 1% of the patients or so in the Stribild arm had some resistance. And when you do have resistance with this combination, you typically see nuke resistance as well as integrase resistance, so dual class resistance. So there's not an improvement on TAF on resistance. It's the same. But there seems to be a safety benefit in renal and bone toxicity. But you're still going to have to select the patients that you feel like are able to take this medication and not have viral failure and then resistance. Okay, this is some brief update on the DCF-TAF. So D means darunavir. So this is, as I mentioned, the first time we'll ever have a boosted protease inhibitor included in a single tablet regimen. So it's darunavir, cobicistat, FTC, and tenofovir alafenamide, all included in a single tablet regimen. And this was compared to the existing darunavir Cobi plus Truvada, which of course is tenofovir DF. And so what you see here, again, the same sort of antiviral effect for these two arms. No problem there. It seemed to be equivalent. But this is the benefit of the protease inhibitors. Again, there's no resistance at failure. So in both the DCF-TAF and the darunavir Cobi arm, when you did have viral failures, it did not result in viral resistance. Again, probably the main reason we still use PIs in the clinic today. And this is just some data that just confirms what we thought about TAF with high concentrations in the lymphocytes. So this is in the PBMCs, you can see the TAF, tenofovir levels were very high compared to the Truvada arm. And in the blood, this is the blood PK, you see the tenofovir arm was 10-fold higher than the TAF arm. So much less tenofovir in the plasma, much more tenofovir in the lymphoid cells. Again, concentrating where HIV lives, relieving the bone and renal toxicity. Okay, let's move on to long-acting agents. So this is a really interesting area because there is a very small segment, I'll say. 85% of the patients are undetectable at Thomas Street, but that 15% really give us fits. And we need some new strategies on them. And these long-acting agents may just be the thing that they need. And so the sort of lead compound on this is a drug called cabotegravir. I always call it carbotegravir, but it's cabotegravir, otherwise known by its acronym 744. It is an analog of dolutegravir. It's available as an oral formulation as well as an IM formulation. The oral formulation has a half-life of 40 hours. The IM injection, 40 days. And so this incredibly long half-life is the basis for giving it as a depot injection. Of course, you can't give it by itself. It needs a partner. And so rilpivirine, actually, the non-nucleoside rilpivirine is now being developed as a long-acting agent as too. And so there's this combination of cabotegravir and rilpivirine that's being developed as a dual drug treatment regimen. So as you know, we've learned through the long sordid history of HIV that you need 3 drugs to treat HIV. Well, here we are going back to 2, and maybe it'll work this time, right? So dolutegravir, rilpivirine, long-acting agents. There's some basis for this. They have different mechanisms of action, of course. There's no drug-to-drug interactions. There's no drug-to-drug interactions with the Hep C meds, so this would be nice for that. And they can do a synchronous injection cycle every 4 weeks or every 8 weeks, still to be worked out. The good thing about these two is there's oral formulations available as well. So you're going to lead in with the oral, or you're going to back off with the oral. The oral will be used to transition off the injection forms of these. So this is the proof of concept. You got to prove to these HIV providers that 2 drugs actually works, because we know that it takes 3. So can we really maintain people with these 2 drugs? Well, this was done with the oral formulations, not the IV formulations. And so you see all these different doses of cabotegravir, and they're starting off with nukes. So we're going with the standard, okay? 2 nukes and cabotegravir, okay? But then 24 weeks later, we're transitioning the patients to cabotegravir and rilpivirine, oral. Okay, these are the oral, because this is a proof of concept that this dual drug regimen can maintain these patients' viral suppression. And it did. Okay, so this is the induction phase, 24 weeks, a bunch of people undetectable. This is the maintenance phase. They stayed undetectable. So in this study, the dual drugs, cabotegravir and rilpivirine, were able to maintain viral suppression over 48 weeks, and then it's been extended to 96 weeks. So it seems like this may work. This is the resistance thing on this, because remember, dolutegravir, a very high resistance barrier drug. Rilpivirine, probably the lowest of all the drugs in resistance barrier. So you got to worry about resistance with this 2 drug agents. In the induction phase, there was 3 patients who had viral failure. None of them had resistance. They probably weren't taking it. In the maintenance phase, you had 2 patients have viral failure. 1 didn't have resistance, but 1 did. And so this 1 patient had an integration mutation and a non-nuke mutation. And this is the great fear of using this, is that you just blew 2 very, very useful classes of agents, non-nukes and integrase. And so when this study was extended to 96 weeks, there were 3 more people who had viral failure, and all 3 of them had non-nuke resistance. None of them had dolutegravir resistance. But this is going to be the sort of hesitation in this regimen, that you're giving these patients these long-acting agents, and you're not seeing them very often, and if they fail, they're going to fail with dual resistance. So this needs to be studied. Now, when we go to the long-acting form, this is new for everybody, okay? So that's why there's this big mess on this slide, because nobody knows how to do this. We don't know the dosing schedule, we don't know the amount or whatever, so they're trying a whole bunch of stuff. But what we see is that the PK looks really good. So this is cabotegravir, okay? And this red is the 800 milligrams every 4 weeks. And you can see that it's accumulating, okay? Nobody knows what the toxic level is, but it seems to be that's too high, okay? What about the green one, 800 milligrams quarterly? And you see that's getting kind of low, because here's 5 milligrams of oral cabotegravir. So the dose is going to be somewhere in here, but we don't really quite know yet, okay? What about rilpivirine? Rilpivirine's shorter. It doesn't have as long half-life as cabotegravir. So here you see rilpivirine. It's going to have to be dosed pretty much every 4 weeks. So rilpivirine is going to need every 4 weeks. Cabotegravir might be every 8 weeks, but isn't that going to confuse everybody? This 4 weeks, this 8 weeks. So they're probably looking at either every 4 weeks of injection of both, or trying to stretch rilpivirine to make both 8 weeks. But it remains really to be seen and developed. So in addition to treatment maintenance with cabotegravir and rilpivirine long-acting, it's also being developed as PrEP. So cabotegravir in itself is going to be developed as PrEP, dosed every 3 months. So that might be very interesting. Come in for your quarterly PrEP shot. Okay, what about new mechanisms of action? This is pretty exciting too. So this is an attachment inhibitor called BMS 626529 with its prodrug 663068. How does this work? Well, GP120, GP41 is it's sort of the hand that HIV uses to grab CD4. And so it induces this conformational change right there on GP120 when it gets close to CD4, and then it binds co-receptor and enters. So remember, Maraviroc, which is the CCR5 inhibitor, blocks here, but this is an attachment inhibitor. It's a new class, a new mechanism of action. How does it work? Well, it attaches, no pun intended, yes, it was intended, it attaches to the conformational site right here that change is going to take place. And so when the HIV tries this conformational change, the CD4 cannot bind because BMS 626529 is sitting in the way. And so this is a new mechanism of action to block attachment. It doesn't depend on co-receptor because it acts before the co-receptor. And so this is very exciting. A bunch of different dose-finding studies. We're not going to sit here and analyze this, but the proof of concept is it works. So this is a dose response curve of this attachment inhibitor, and you can see over a 7-day monotherapy dosing, it has about a 1.5 log decline in HIV. If you put it in with a combination therapy, about 80 to 90% of the patients become undetectable at 48 weeks. So very exciting, new mechanism of action, attachment inhibitor. And also at the recent meeting in Vancouver, BMS presented data on a maturation inhibitor. So the very beginning, the attachment, and the very end, the maturation, we might have 2 new mechanisms of action. So we're still evolving strategies in HIV. Okay, let me finish with cure and then we can all go home. So I'm not a basic scientist, I'm a clinician, and so I don't understand all this stuff, but I think it's pretty cool. So the cure strategies are, one is you could vaccinate somebody before they were exposed, so that would be a cure, I guess. Very early treatment to limit the viral reservoir. So if you could, it's like sepsis, if you could just give those steroids right before they got exposed to E. coli, boy, you could do a lot. But how do you know when they're going to get exposed? Same thing with HIV. If you could just treat them right before they got infected or right after, but we don't identify people that early. Viral clearance by the immune system, the development of therapeutic vaccines. Listen to this, transfusing cells without CCR5. So you actually do gene editing and cord blood transfusion. So I'm really glad we saved the cord blood on my kids. Actually, it's out in Phoenix somewhere. But they can edit the genes and actually transfuse and to convert the patient to have CCR5 negative cells. And so that's actually how the only patient that was ever cured of HIV was cured. He had a stem cell transplant from a patient donor that had CCR5 mutation, and he got cured. And so they're trying to edit a patient's own genes to self-cure them. And then the last one is this reactivation of the infected latent cells. So sort of we have these infected cells that are just sitting there latent that last forever, and if we could just shake those out, then we could cure someone of HIV. And so this is called the kick and kill strategy. So you have these latently infected cells, and you kick them with either histamine deacetylase or protein kinase C agonist, or as I'm going to show you in just a second, toll-like receptor 7 agonist. You make the cell express HIV proteins, and then you kill it, either with a therapeutic vaccine or these other strategies. And so you're basically trying to flush out the reservoirs. And this is the last slide. It's an actual a glimpse of a trial in this, which is Gilead 9620, which is a toll-like receptor 7 agonist. So it basically is the kick agent, and then you're going to kill the cell that's expressing these HIV antigens. And so this basically shows from all of these patients, 18 patients, this is ex vivo, so they take PBMCs from these 18 patients, and they kick them. And what you can see is that the fold HIV activation is quite strong in most of the patients, but not so much in some of the patients, so they got a lot of work to do in this. But it is a proof of concept that you can kick these latent cells, make them express HIV, and then use something else to kill them. So with that, I'll finish, and I want to thank you all for hanging in there to the bitter end, and thank you again, Harris, for including me. I appreciate it.